Welcome everyone, I'm Weyler from the University of Bonn, and in this talk I want to present our work about joint plant instant detection and leaf count estimation for in-field plant phenotyping, in which we implemented a vision-based approach to localize crops and weeds in the field and simultaneously estimate the total leaf count for each plant to assess its growth stage. The motivation for our work comes from the areas of precision farming and plant breeding, in which the assessment of the growth stage of individual plants plays an important role. In the context of precision farming, knowledge about the growth stage is relevant for targeted management actions performed by agricultural robots on the field. For example, certain herbicides and fungicides will be most effective when applied at a certain growth stage. On the other hand, plant breeders are continuously generating new genetic variations of crops to keep the yields high. These variations are then planted and their performance is assessed. A key element to assess the performance and the vegetative development of individual plants is to analyze their growth stage, which can be described by the BBCH index. This index is mainly defined by the total number of leaves developed at the main stem. In our approach, we developed a vision-based model which is applicable to real-world field data acquired by agricultural robots and enables us to localize crops and weeds in the field by predicting their corresponding bounding boxes. Simultaneously, we estimate the location of plant-specific leaf key points and associate them to individual plants on the field, which finally allows us to estimate the total leaf count at a plant level. However, assigning individual leaves to a specific plant is challenging due to overlapping plants, as is often the case in later growth stages. For this task, we train a deep neural network which predicts the bounding box of each plant by estimating the location of its top left and bottom right corner via heat maps. At the same time, we predict the location of plant-specific leaf key points. To associate each corner and each leaf key point to an individual plant on the field, we additionally predict embeddings and train the network with the objective of small distances between all embeddings which belong to the same instance, shown here by identical colors, and large distances between embeddings of different instances. Thus, we can associate a leaf key point to its corresponding plant even in case of overlapping plants. We compare our model with a two-stage mask Gaussian N approach and show that our model achieves a more accurate total leaf count with a higher precision. Finally, I would like to give a short summary. We developed a model to analyze plant development in complex scenes on real agricultural fields. First, by localizing crops and weeds using a single-stage object detection approach, and simultaneously we detect plant-specific leaf key points which we associate to individual plants on the field. This enables an automated leaf count estimation for large-scale phenotyping. With that, I would like to thank you for your attention.